Unit 4 of your general biology course will cover the cell cycle, cell division, and a little bit over human reproduction. The information will be coming from chapters 9, 10, and a small portion of the information can be found in chapter 36 in your textbook, Campbell Biology in Focus, which is in the second edition. There are several handouts, including one called Cell Cycle and Cell Division Diagrams, and videos that are included in Canvas that should be very helpful for you for this unit. Let's begin by taking a look at the cell cycle. And the cell cycle is essentially a sequence of events that a cell will go through as it proceeds through the process of dividing. And remember, to kind of keep it simple here in terms of what is happening, essentially we're taking one cell and we're going to make two in the case of mitosis. If we're talking about meiosis, in most sexually reproducing organisms we will go from one cell and go through two divisions of the nucleus and actually end up with 4 daughter cells. But regardless of the type of cell division that we're talking about, the whole process can kind of be illustrated through this diagram, which is called the cell cycle. And the cell cycle is made up of 4 phases. Those 4 phases are G1, S, G2, and the M phase. If you look closely, G1, S, and G2, those 3 phases combined are all called interphase, and that's kind of preparation for cell division. During G1, S, and G2, the cell is not actually going through the process of division, but it's just preparing for cell division. And this takes approximately 90% of the time of cell division. So if we're talking about a 24-hour period of time for a cell to divide, then approximately 20 hours of that time is actually spent in those first 3 phases, G1, S, and G2, where the cell is not technically dividing, it's just making preparations for cell division. And those first 3 stages are called interphase. If we look at each one of the phases, we can see that there are specific things that are occurring. So for example, the G1 phase, which your book will refer to as the gap phase, I tend to think of it as a growth phase, because again, this is when the cell is kind of going through these leading up processes for cell division. And during G1, basically the cell is actually going to increase the number of organelles. And so if we're going to go from 1 cell to 2 cells, for example in mitosis, then both of those daughter cells are going to need the organelles necessary to function. So we're going to need more mitochondria, more ribosomes, more of the endomembrane system, the smooth and the rough ER and the Golgi apparatus, so that both cells have an adequate number of organelles to function. Also during G1, we're going to increase the volume of cytoplasm, which contains many of the important nutrients that the cell will need for an energy source, and other proteins to function. The S phase is called the synthesis phase, and this is when DNA is going to replicate. So for example, in the nucleus of your normal body cells, your somatic cells, you have 46 chromosomes. During the S phase, we're going to replicate or duplicate each one of those chromosomes. So for a short period of time, you'll actually have 92 chromosomes in the nucleus of your cell. That's to ensure that when the cell divides, that both daughter cells will have a complete copy of the DNA, the information they need to drive the cell's chemical reactions. G2 is the second gap phase, and this is basically just final preparations for cell division. So any last minute things that need to be taken care of in terms of the cell dividing will occur during that G2 phase. So additional proteins, enzymes, any structures that the cell will need to complete cell division will be taken care of during that G2 or gap phase 2 of the cell cycle. The M phase is actually technically mitosis, and so this is when we're going to physically start the process of cell division, and this takes about 10% of the time. So G1, S, and G2, those three combined are called interphase, and then the M phase or the mitotic phase takes 10% of the time, while G1, S, and G2 take approximately 90% of the time. The M phase can be divided into those traditional phases of mitosis that you've heard: prophase, metaphase, anaphase, and telophase. Let's just do a little bit of a comparison and contrast between the two types of cell division. The two types of cell division are mitosis and meiosis. Let's start with a specific type or a special type of cell division that occurs in sexually reproducing organisms called meiosis. And meiosis really is the basis of sexual reproduction. So meiosis occurs in the male and the female sex organs. So in the male, that's the testicles, and in the female, that's the ovaries. And we'll kind of look at the timing of this a little bit as we get further into this section. But the result of meiosis basically are these specialized reproductive cells called gametes. And the gametes in the male are the sperm cells, and the gametes in the female are the egg cells. And really the purpose of meiosis is to reduce the chromosome count by 1/2. And so again, if we think about our own cells, our normal body cells or somatic cells, the normal or diploid chromosome count is 46. In our gametes, we'll have 1/2 of that diploid chromosome number, which is called a haploid chromosome count. And so in the gametes, in the sperm and the egg, we'll have 23 chromosomes. And this kind of side-by-side comparison shows you what happens with these chromosome counts. If you look on the left-hand side, starting with the parent cell and kind of moving over to the left, you can see that we start with a 2N or diploid chromosome count number of 6, and then when we look at the two daughter cells, we end up with the same number of chromosomes. And so essentially what we do is we've replicated those chromosomes, and then we've divided that genetic material just once. And so we go from a diploid chromosome count of 2N equaling 6 to a diploid chromosome count of 2N equaling 6 in the two daughter cells. We say the chromosome count is preserved in mitosis. When you contrast that with meiosis, we start out the same, so a diploid chromosome count of 6. We replicate the genetic material. So in this scenario, for a short period of time, we actually have 12 chromosomes, and then we divide the genetic material twice, and so they call that meiosis 1 and meiosis 2. But if you follow that parent cell over to the right and kind of look through the process, you'll see that there's 2 divisions of the genetic material, and we end up, instead of 2 daughter cells, we end up with 4 daughter cells, and those 4 daughter cells have a haploid chromosome count, which is represented shorthand by N. And that haploid chromosome count in those 4 daughter cells is 3. So we've reduced the chromosome count by 1/2. If you see, and we'll talk about those terms here in a little bit, but if you see that 2N, that represents a diploid chromosome count. Di means 2. That basically means you have 2 copies of the same type of chromosome. N represents the haploid chromosome count. That means that you have just 1 copy of that particular type of chromosome in those cells, and you get that haploid chromosome count in the gametes. So this goes into a little bit more detail, and you can kind of see this haploid diploid scenario playing out in the graphic on the left. As we mentioned earlier, there are basically 2 types of cells. that you have, and those are your normal body cells or the somatic cells. Those are things like your skin cells, your nerve cells, your muscle cells. They have that diploid chromosome count. And then the specialized cells in sexually reproducing organisms, those reproductive cells are called the gametes, the sperm and the egg, which have that haploid chromosome count. And so essentially we start in the reproductive organs with meiosis in the testicles and in the ovaries, and we begin with the parent cell, and we are going to basically replicate the DNA, but then we go through 2 divisions of that replicated DNA and we reduce the chromosome count by 1/2. And so we go from a diploid to a haploid chromosome count in the gametes. The normal chromosome count, the diploid chromosome count, basically is restored during fertilization. And so when you get the fusion of the sperm and the egg, both of those being haploid, the first cell that results is called the zygote, and that zygote is diploid. Again, represented in shorthand by that 2N notation. This just shows a human karyotype. And again, we have a total of 46 chromosomes, but those 46 chromosomes occur in 23 pairs. They're called homologous pairs. Essentially, they're carrying the same type of information with respect to the genes that they are carrying. And remember that those genes are sequences of nitrogenous bases that code for specific proteins that are expressed as certain traits or characteristics, hair color, eye color, all of those physical traits that we see expressed in an organism. The chromosomes, if you look at the karyotype, they're kind of color coded there for you, and this doesn't show all of the genes, but that's meant to represent different areas of the chromosome that would have specific information for a gene that's carrying the essentially the sequence of information for building the proteins that code for specific traits or characteristics that we see expressed in the individual. All of the chromosomes, the chromosome pairs 1 through 22, if you look at them, they're kind of very similar in terms of their homologous pair, similar size and color coding and all of that business. But then if you look at the last pair, which would be the 23rd pair, it's not numbered 23. It shows up here as a X and a Y. That is actually the gender chromosomes, and that determines male or female. If it's XX, then that individual is female. If it's XY, then the individual is male. So this particular karyotype is a karyotype of a male. Let's look at mitosis for just a minute. And so we're going to focus over here on the left-hand side of the diagram. Mitosis is kind of just this normal type of cell division, and it occurs in your somatic or your body cells. And this is basically going on all of the time. And so your skin cells, your blood cells, your muscle cells, up until you reach a certain point of maturity, are kind of going through this process of mitosis or cell division. And basically what happens, it's really just kind of it's done for several different reasons, but one of the reasons is it's just kind of basic housekeeping. And so we need to continuously replenish our cells as they break down and wear out. But what happens during mitosis is we're going to replicate the DNA. And so we start out with a diploid chromosome count, in this scenario at 6. We replicate it for a short period of time, we have 12. Then we divide it between the 2 daughter cells, and then the 2 daughter cells are also diploid, or you have 2 copies of the same type of chromosome. In mitosis, we say that the chromosome count is preserved. We'll talk about some other reasons here in just a little bit about why a cell would go through this process of mitosis. With regards to the processes that are involved in cell division and in mitosis, there really are two, and that involves the division of the nucleus, which is called karyokinesis. So we have to divide the genetic material, but we also have to divide the cytoplasm between the two daughter cells. Karyokinesis is the division of the nucleus. It basically forms a nuclei for each of the two daughter cells. It begins in prophase and it actually ends in telophase. And so all of that lead up to cell division, G1, S, and G2, we're not technically starting the process of mitosis yet. That begins in prophase when we start the division of the nucleus, which is called karyokinesis. And then the other major process that's involved in cell division is cytokinesis. And this is the division of the cytoplasm. It looks a little bit different with regards to how it's accomplished and what structures are present, depending on whether or not you're talking about a animal or a plant cell. We'll kind of compare and contrast cytokinesis in plant and animal cells here as we move forward. But for the most part, cytokinesis is a division of the cytoplasm, and it basically, especially in mitosis, it ensures that you have an equal division of the cytoplasm and you get two daughter cells of equal size. It's a little bit different in meiosis in the female, and we'll see that as we look at oogenesis and meiosis in the female. Before mitosis can proceed, remember that we have to go through those first three stages of the cell cycle, G1, S, and G2. But during the S phase of the cell cycle, DNA is going to replicate. And we looked at DNA replication in the previous unit, where we kind of saw this process of the replication bubbles forming, the separation of the duplicate strands of the DNA molecule, and then the building of the complementary nitrogenous base pairs for both parent strands, and so this kind of represents that process in a little bit more detail. Remember, this is occurring during the S phase of the cell cycle. And what happens once DNA has replicated, essentially there are two forms of DNA. There's what's called chromatin, and chromatin is kind of the loose, unconsolidated form of the DNA. And then the other form that you can find DNA is in the form of chromosomes. So once that DNA is replicated, it starts to tightly coil and condense and form these duplicate chromosomes. These duplicate chromosomes form pairs, and when you see two of these duplicate chromosomes together, those pairs of chromosomes are called chromatid or sister chromatid. And the sister chromatid basically are held together by this little constricted region called a centromere. So this is all, again, part of leading up to the process of cell division, be it either mitosis or meiosis. Essentially, one of the things that has to happen after we increase the volume of cytoplasm and the number of organelles is we have to replicate the DNA. Once that DNA is replicated, it starts out kind of in the chromatin form, then it kind of becomes tightly coiled and condensed and forms these chromosomes, these duplicate chromosomes, which we will refer to as sister chromatid, and they're held together by that little constricted region called a centromere. You want to be careful. There's a lot of vocabulary in this unit, and a lot of those terms sound very similar, and so pay close attention to the vocabulary as it's introduced. We're going to go through the phases of mitosis, and we'll actually start with the preparatory phase or interphase, which technically is not mitosis. It's just a lead-up process, or the cell kind of making preparations for cell division. I always tend to think of interphase as being a part of this bigger process of cell division. You can't really have mitosis without interphase. And so interphase, if we kind of think back just briefly, constitutes the first three phases of the cell cycle, G1, S, and G2. Takes 90% of the time. And if you are looking at a group of cells, for example, the one over on the right hand side near the top of the slide, this is a microscope image, probably, I don't know, somewhere around 800 times magnification. And these are cells that you would find in an onion root tip. And this is a longitudinal kind of section of an onion root tip. The cells have been stained and with a differential stain, so you can see the nucleus and the nucleolus and the chromatin and the cytoplasm, the plasma membrane, and the cell wall. And so it's important to be able to sort of recognize or identify the structures that are present if you want to be able to determine what phase of cell division that a cell might be in. So those are things that you'll be looking for. The nucleus and the nucleolus are visible. Now the graphic on the upper right hand side is a graphic of plant cells, and the plant cells tend to be kind of these uniform size and shape. The one over to your left, both the microscope image that's been color enhanced and also the kind of the little diagram below are meant to represent more of like an animal cell. And you can see the nucleus and the nucleolus present, and the DNA basically, and they're showing it as the chromosome form. To me, this is still kind of in the chromatin form, leading to these tightly coiled condensed chromosomes. You'll see that term centrosomes associated with the centriola pairs, which are all part of these protein structures that will play a role in cell division. An interesting thing about the centriola pairs is we don't see those in plant cells. We only see those associated with animal cells. Although there's an analogy, there's a similar structure that performs a similar function in plant cells, but centriola pairs would be found only in animal cells and not in plant cells. Make sure you're kind of paying attention, especially to these graphics, this figure 9.7. These graphics, you'll probably see examples of these. They're included on that handout that I referenced earlier over the cell cycle and cell division diagrams. They're in your textbook in chapter 9, and you'll also be expected to be able to identify certain structures and the phases of mitosis related to these figures. So take a close look at those as you get a chance. After interphase, and again, which is the first three stages of the cell cycle, we move into the first phase of the M phase of the cell cycle, which is prophase. And there's a lot going on during prophase. Your book actually will break this down into prophase and prometaphase. Don't let this be confusing for you. I'll just refer to it as prophase. But remember that this is a continuous cycle, and as you kind of look at these different cells, sometimes it might be a little bit difficult to distinguish if you're talking about late interphase, early prophase, or late prophase, early metaphase. It's kind of a continuum or a continuous cycle, but we're looking for certain kind of signposts or traits or characteristics during each one of these phases to identify the cell with regards to where it is in this process of cell division. But by prophase, DNA is replicated again during the S phase of the cell cycle, and that DNA is kind of going from that loose unconsolidated chromatin form to the chromosome form, and these duplicate chromosomes, which we saw earlier, that are referred to as sister chromatid. During prophase, this is the beginning of that first major process of cell division called karyokinesis. And so the nuclear membrane will start to break down. And that signals the beginning of karyokinesis. Karyokinesis is going to begin in prophase. These centrosomes, which are these protein complexes with the associated centriola pairs, again in animal cells, we'll start to see the formation of these spindle fibers. And these spindle fibers are basically these protein strands that are going to attach to the centromeres. The centromeres are those little constricted regions that hold the duplicate chromosomes together. As the centriola pairs and the associated spindle fibers attach to the centromere and the nuclear membrane begins to break down, those centriola pairs with their spindle fibers start to migrate towards opposite poles. As they start to move towards or migrate to the opposite poles, that's going to have an effect on those duplicate or sister chromatid. The effect that the spindle fibers have as they kind of move towards these opposite poles is that they force or push those duplicate chromosomes, those sister chromatid, basically to the middle or the center or the equator of the cell. So this really signals the next phase of mitosis as we transition from prophase into metaphase. Those duplicate chromosomes are going to be forced to or lined up through the middle or the equator of the cell. By this time, the nuclear membrane is completely gone, and the chromosomes are being moved towards or lined up through the middle or the equator of the cell. Think of meta as middle, and metaphase I think is probably one of the easier ones to identify. And again, you can see the graphic on the right hand upper portion of the slide, which illustrates basically metaphase in a plant cell. And then over to your left, the color enhanced diagram, the kind of the fluorescent blue structures indicate the sister chromatid or the duplicate chromosomes through the middle or the equator of the cell. The yellow protein strands are the spindle fibers that have attached to the centromere, which have kind of forced those chromosomes to the middle. That combination of the centrosomes, the spindle fibers, the centriola pairs in animal cells, they'll all refer to as the spindle. For me, I tend to think of those protein strands just as the spindle fibers, and you'll hear me use that terminology a little bit more than just referring to that whole structure as a spindle. As we kind of move from metaphase to the next phase of mitosis, which is anaphase, the thing to look for is the separation of the duplicate chromosomes. And so in the back of your mind, remember what's happening here. Basically, is we're taking one cell and we're going to, in the case of mitosis, make two cells. And anaphase is the first phase of mitosis where you can actually see the separation of the duplicate chromosomes or the sister chromatid. And basically what they're doing is they're following those spindle fibers back to the opposite poles of the cell. And there's different mechanisms for the separation of those sister chromatid. Essentially the spindle fibers kind of act as a track, and now what are the individual chromosomes basically are going to follow that track back to the opposite poles as they move away from one another, and eventually will become individual chromosomes in the daughter cells. And so anaphase is the first time that we see the separation of the sister chromatid or the duplicate chromosomes as they begin to follow that those spindle fibers to the opposite poles of the cell. Remember that this is a cycle, and there's kind of a continuum here, and so we're looking for certain traits or characteristics to determine, are we talking about prophase, metaphase, anaphase, or telophase. And one of the things that really distinguishes telophase from anaphase is that the individual chromosomes now basically have moved towards to the opposite poles of the cell, and the nuclear membrane is going to start to reform. And as those individual chromosomes basically start to reform these daughter nuclei, they're going to go back from that tightly condensed or consolidated chromosome form back to the chromatin form. A centriola pair in animal cells will stay with each of the reforming daughter nuclei, and this really is kind of the end of karyokinesis, that first major process in cell division, and we're finishing up the process of dividing the nucleus, but it's the beginning of cytokinesis, the division of the cytoplasm. And one of the major differences between plant and animal cells is how we divide that cytoplasm, how cytokinesis is accomplished. If you look at the graphic in the upper right hand side of the slide and you look closely, you can kind of see these dark striations or bands between the two daughter nuclei. Those represent the spindle fibers, but then running horizontally there's kind of a light tannish band that is cutting through the middle of these two daughter cells. That's called the cell plate. And that formation of the cell plate is kind of a combination of the plasma membrane and the cell wall. It starts to grow out from the middle of the cell and divide the cytoplasm. And so that's the beginning of cytokinesis in plant cells. In animal cells, you get a pinching in basically of the plasma membrane between the two daughter cells, and that membrane just continues to pinch in until eventually it completely divides those two daughter cells. That little indentation or pinched in region of the plasma membrane is referred to as the cleavage furrow. Well, that's one of the major differences in terms of cell division in plant and animal cells. So telophase kind of continued. We finished up karyokinesis. Cytokinesis begins. In animal cells, basically the plasma membrane is going to pinch in and divide the cytoplasm between the two daughter cells, creating that little indentation called the cleavage furrow. In plant cells, a cell plate is going to form. Remember that this is occurring during telophase. That cell plate consists basically of the plasma membrane and the cell wall. It kind of starts in the middle of the two cells and grows outward and divides the two daughter cells, resulting in these two diploid daughter cells. Remember, the chromosome count is preserved, and we're talking about mitosis in this scenario. Now these two daughter cells basically can independently go back into the cell cycle and enter G1 and go through G1 and S and G2, which are all part of interphase, and then if depending on the type of cell, it may continuously stay in the cell cycle. Some cells will kind of continuously divide if it's a plant cell, and if it's part of the meristematic region where the plant is actually is actively growing, then those cells will almost continuously divide. Cells like your epithelial cells, your skin cells that are kind of constantly being sloughed off and need replaced, they'll kind of continuously stay in the cell cycle. Other cells, especially things like your striated muscle cells, once you're mature, they really won't divide anymore. Bone cells, those types of things, when you're actively growing, will go through cell division, but at a certain point they will start to slow down or tail off in terms of dividing. This graphic just kind of shows you in a little bit more detail the differences between cytokinesis and in animal and plant cells, with the formation of the cleavage furrow as contrasted with the formation of the cell plate in plant cells. And that kind of wraps up the first part of this over the cell cycle, and just kind of background information on some of the vocabulary and terminology that you need: haploid, diploid, chromosome counts, comparing and contrasting meiosis, and then a closer look at mitosis. Here you can see the cell cycle. They do go ahead and break that mitotic phase down and add that additional phase of mitosis. So whereas I will basically just talk about interphase, prophase, metaphase, anaphase, and telophase, they kind of stick that pro-metaphase in there. That's something that's fairly new, and we'll just kind of stay with those traditional phases of mitosis and don't let that pro-metaphase of mitosis in there confuse you. It's just meant to illustrate really a transitional phase between prophase and metaphase. We're going to wrap up here with a short overview of the other type of cell division, which is meiosis. And again, meiosis is this special type of cell division that occurs in sexually reproducing organisms. And meiosis, a couple of things in general to keep in mind is we're going to talk about the sequence of when this occurs, where it's happening, and the location. We're going to look at meiosis in humans and kind of talk a little bit about meiosis in the male, spermatogenesis, and meiosis in the female, oogenesis. And I would strongly encourage you to take a look at those videos along with the diagrams that are provided to you in Canvas. I think going through the process of looking and doing those drawings for all of the different components that we've talked about with respect to cell division, the cell cycle, mitosis, and then meiosis, and meiosis specifically in the male, spermatogenesis, and meiosis in the female, oogenesis. I think doing those drawings and looking at those diagrams will really help you kind of put the pieces together. But meiosis is cell division in the reproductive organs. It produces these special sex cells or reproductive cells called gametes, sperm in the male and ova or eggs in the female. The chromosome count in the gametes is haploid. It's represented in shorthand by just the letter N. Is compared to the chromosome count in our normal or somatic cells, in which the chromosome count is diploid, represented in shorthand as 2N. In your somatic cells, your normal or diploid chromosome count is 46. The haploid chromosome count in your gametes and the sperm and the egg is 23. One of the major differences between meiosis and mitosis is that we actually are going to go through two divisions of the genetic material. We're going to go through two divisions of the nucleus, essentially. Those two divisions are called meiosis 1 and meiosis 2. And the end result is we go from, it starts out the same. Basically, we start out with a diploid chromosome count, or two copies of the same type of chromosome, but one of the major differences between mitosis and meiosis is what results. And so after mitosis, we just get 2 daughter cells and we go from the diploid to diploid chromosome count. In meiosis, we go through 2 divisions of the genetic material, and instead of 2 daughter cells, we end up with 4 daughter cells that are haploid, or have half of the normal chromosome count, or just 1 copy of each type of chromosome. The purpose really is to reduce the chromosome count by 1/2 in the gametes, and so that the normal chromosome count can be restored during fertilization. And that first resulting cell after you get the fusion of the gametes, the sperm and the egg, is referred to as a zygote. If you follow this back and you look at it from an evolutionary perspective, we've talked about 2 basic types of reproduction: asexual reproduction and sexual reproduction. Asexual reproduction kind of more in bacteria, algae, protozoans, some of these what we would consider to be maybe less complex organisms. And the upside to asexual reproduction is just a numbers game. So they can get a lot of individuals out there in a short period of time. The advantage to sexual reproduction really is genetic variability, because we are a combination, and sexually reproducing organisms were a combination of genetic material from 2 different individuals. That gives us genetic variability. That genetic variability is a big deal. And it's not to say that asexually reproducing organisms don't get genetic variability. We know they do through transformation, transduction, conjugation. But in sexually reproducing organisms, that really is the evolutionary advantage. And because the vast majority of organisms on the planet do sexual reproduction, and we look for these reasons from an evolutionary perspective, well, why would that be? Well, the reason is genetic variability. And genetic variability improves the overall health or fitness of a population. So if you have genetic variability and something changes in the environment, then some members of that population just purely by chance and due to that variability in the population are going to have a competitive advantage. Those individuals are more likely to survive. They're going to reproduce, and they're going to pass on those traits or characteristics that have given them that competitive advantage. That's all part of the evolutionary process, specifically natural selection. So anyway, let's kind of continue on with this conversation about meiosis. Meiosis in the male is called spermatogenesis. It occurs in the reproductive organs, and the reproductive organs of the male are the testicles. The testicles made up of 2 parts basically: the testis, where the sperm is made, and the epididymis, where the sperm is stored. Spermatogenesis begins at puberty, basically with an onset or change in hormones, and in the male, male meiosis, spermatogenesis, basically continues throughout the course of the male's lifetime. As the male gets older, the quality and the number of sperm will start to tail off a little bit. It's just kind of a natural process called senescence. But essentially the male will produce sperm throughout the course of their entire lifetime, and literally produce hundreds of millions of sperm cells over a 24-hour period of time. The sperm is basically stored in the epididymis until it's released from the body. If it's not released from the body, then essentially it breaks down and is recycled. And so the specific type of meiosis in humans, in the male, is called spermatogenesis. Again, just a couple things to remember. There's 2 divisions of the genetic material, meiosis 1 and meiosis 2. It starts with the parent cell called the spermatogonium, and through the 2 divisions of the genetic material, we go from a diploid chromosome count to a haploid chromosome count in the gametes. And again, the gametes in the male are the sperm cells. Let's take a little bit closer look at meiosis in the female. Meiosis in the female is called oogenesis, and it occurs in the reproductive organs. The reproductive organs in the female are the ovaries. The timing of meiosis in the female is a little bit more complicated, I think, than the timing of meiosis in the male, in that oogenesis actually begins in the embryonic ovary. And so this process of oogenesis starts while the embryo is still in the womb of the mother. It begins with the parent cell, the oogonium, and continues to a point with the first cell that results from the oogonium called the primary oocyte. Basically, the human female has all of her eggs in the ovary in an immature state, essentially at birth. And so this process of oogenesis will continue to a certain point in the embryonic ovary, and then it's stopped or arrested basically at birth, and doesn't pick up again until the onset of puberty with hormonal changes, estrogen and progesterone, and other hormones. Well, if you look, you can see that after meiosis 1, this looks a little bit different than the results from meiosis 1 in the male in spermatogenesis, where we had basically 2 cells of equal size. In the female, we get what's called unequal cytokinesis. Remember that cytokinesis is basically the division of the cytoplasm. And with this onset of puberty, oogenesis is going to start up again. When it starts up, basically the timing for the release of the gamete or the egg is on approximately a 28-day cycle. And so every 28 days approximately the female will release one mature ovum or egg. This, if you think about it, makes sense with respect to how the human female and humans in general, the evolutionary path that they have taken with respect to reproduction. And so humans, and the human female body really is only designed to carry one offspring at a time. And so this 28-day cycle, the female will not release 4 mature ovums or eggs because there would be this potential for, basically multiple offspring, which would be very, hard, or could cause problems for both the mother and the offspring in terms of the physical stresses of carrying multiple offspring. And so because humans have followed this evolutionary path where we have very few offspring, and we invest a tremendous amount of parental care in that offspring, a long gestation period, 9 months, there needs to be a way to reduce the chromosome count, but not release multiple gametes per cycle, per this 28-day cycle. And so the way that that's accomplished is we reduce the chromosome count through 2 divisions of the genetic material, meiosis 1 and meiosis 2. But during the division, basically of the nucleus, both of these structures will get an equal number of chromosomes or genetic material, but one of these will get the lion's share, or the vast majority of the cytoplasm. It's called unequal cytokinesis. The other structure gets very little cytoplasm, but does get half of the genetic material. That's not a viable cell. That's called a polar body. And that polar body basically just breaks down and is reabsorbed by the female. The other structure becomes a secondary oocyte. And that secondary oocyte will undergo meiosis 2. And so during meiosis 2, again, we get unequal cytokinesis and we get the formation of the second polar body. And after meiosis 2, basically, we are down to the haploid chromosome count. If the developing ovum or egg is fertilized by a sperm, the gamete from the male, then you get the fusion of the gametes, the sperm and the egg. That first cell that results after the fertilization is called the zygote, and that restores the diploid chromosome count. And then that zygote, that cell, essentially re-enters the cell cycle. And so it just goes through multiple mitotic divisions. You go from 1 cell to 2 cells, 2 to 4, 4 to 8, and so on and so on. And that's basically how we develop into this, magnificent multicellular organism with trillions of specialized cells. And so that's kind of how these processes of meiosis and mitosis are connected in sexually reproducing organisms. If the egg isn't fertilized, basically it's sloughed off during the normal processes of the menstrual cycle. And one of the things to be aware of that's different with respect to oogenesis and spermatogenesis is that in the female there is another change, hormonal change, that occurs later in life, approximately 50 years old. It's variable, where you have hormonal changes that will stop the release of the egg, and that's called menopause. Basically, it's a way to help ensure the health of the mother and the offspring. So obviously a tremendous physical burden to carry an offspring to term, and so as a female matures, maybe is not quite as strong physically and may not be able to support the rigors of carrying an offspring to term, that menopause kind of helps to ensure the safety of the mother and the offspring. And so that concludes the PowerPoint notes for Unit 4. Again, I think this would be a very good unit to take a look at the other videos and support diagrams and worksheets that are available to you in Canvas.